Dear colleagues, my name is Professor Natalia Linkova. I am the head expert consultant of Peptides company. Today the theme of our lecture is Peptides, a new technology for activating the growth of indoor plants and seedlings. Plants bring peace, coziness and comfort to the house, which is so necessary in the hustle and bustle of urban life. Indoor plants are a piece of nature in urban homes that can make us happier, less anxious, minimize negative emotions, and reduce insomnia. In the offices of some large companies, plants are specially added to the interior so that employees can relieve stress and saturate the body with oxygen. Indoor plants for wellness. Indoor plants reduce blood pressure and heart rate by reducing stress, fatigue and anxiety. Indoor plants purity indoor air from toxins and pollutants, saturate it with oxygen. Activate creativity and helps you focus on the task you perform at work. Improve mental health, create a cozy and comfortable environment in the room, and have healthy properties. Vegetable grow is not only a way to get fresh and healthy vegetables, but also a tool for maintaining mental health and finding harmony with oneself and nature, stress tolerance and relaxation. Planting seedlings and caring for plants help reduce stress levels. This is due to the physical aspects of gardening, being outdoors, being physically active, as well as the emotional ones, nature and natural antidepressant. The next one, a feeling of happiness. Interaction with plants promotes the release of endorphins, hormones of happiness, which leads to an improvement in mood. Success in growing and caring for plants strengthens a sense of self-esteem and self-confidence. Taking care of seedlings requires constant attention, which contributes to the development of responsibility and determination. Those qualities are transferred to other areas of our life and help to cope more efficiently with everyday tasks. So, peptides is a natural regulator of plants' growth and vital activity. Function of peptides molecules in plants. The first group of functions is regulatory function. It's regulation of proliferation or division and growth plants, regulation of differentiation of maturation of plants, regulation some signals way of plant cells, and stress response. The next part of regulation is protective action of peptides on plants. It's protection against pathogens and increased immunity of plants. Peptides are a popular research subject to the field of crop production and antimicrobial substances, plant growth regulators, and insecticide and herbicide activators. In legumes, clay peptides regulate the endosymbiotic relationship between nodular bacteria and the host plant. Peptides contribute to the accumulation of nitrogen from complexes which metal cations and affect the bioavailability for plants. The physiological action of phytohormones such as auxins, cytokines, gibberellins, and others which control plants' growth and development is potentiated by peptides. So on this slide you can see the effect of the Ka peptide, the active principle of ladonics, and the IEDG peptide, the active principle of endoluten, on the growth and development of the callus culture of the Tobacco nicotiana plant. Ka and IEDG peptides regulate the growth, development, and differentiation of callus culture of tobacco plants Nicotiana tobacco. They increase the growth of callus mass, stimulate the formation and growth of leaves in microplants. Ka and IEDG peptides modulate the expression of some groups of genes. Cle family encoding endogenous regulatory peptides responsible for the proliferation and functional activity of plant cells. The Knox family regulate of transcription of genome of cells, and the GRF family of growth factors regulate encoding DNA binding proteins such as topoisomerase and nuclease. On this slide you can see the effect of the IEDG peptide on the expression of GRF growth factor in tobacco plants. We can see that IEDG peptides can double the expression of the GRF4 gene and increase the metabolism of tobacco plants. GRF is a transcription factor that stimulates the activity of other genes involved in nitrogen uptake and assimilation. The GRF protein coordinates the uptake of nitrogen from the soil by plants, which the uptake of carbon from the atmosphere. Also we can see the effect of IEDL peptide active principle of tax arrest on growth of tobacco plant seeding. Addition of the IEDL peptides to tobacco microplants with increased salt natrichlorid contents leads to a decrease the oxidative stress of cells and a decrease in the toxic effect of natrichlorid. When the IEDL peptides is added to plant cells, it is localized mainly in the endogen zones and root hairs, to a lesser extent in the meristem zone. In the presence of the IEDL peptide, the expression of the VOX7 gene decrease, resulting in the activation of stem cell differentiation and root elongation. So in other experiment we can see the effect of Ka peptide and IEDG peptide on growth, development, and yield of soybeans. Pre-treatment of soybean seeds 1 month before planting with peptide Ka and IEDG at various concentration had a positive effect on the development and yield of soybean, despite the uneven distribution of precipitation and unstable temperature condition. In a handmade experiment, the use of Ka peptide increased soybean yield by 59.81% and the use of the IEDG peptide by 63.84% compared to the control. The use of the Ka peptide in a production experiment contributed to an increase the number of soybeans by 69% and an increase in yield by 30.5% compared to the control. The use of the IEDG peptide in a production experiment contributed to an 88% increase in the number of soybeans and at 18.4% increase in yield compared to the control. On this slide we can see that Ka peptide increase productivity in soybean yield by 30.5%. Also, IEDG peptide increase number of beans production by 88%. In the other experiment, we can see effect of Revilab SL09 on the growth and development of Calenus and Petunia under stress condition, freezing. Revilab SL09 contains IADG peptide, other short peptides, and plant extract. On average, all 10 cuttings of Mother Calenus bush Petunia variety died in the control groups after frosts. Under the influence of Revilab SL09, 2 drops per half liter of water, 3 out of 5 cuttings died and 2 took root. The top photo clearly shows the development of the root system and green leaves in the plant. Petunia cuttings also root poorly after frosts. Under the influence of Revilab SL09, also 2 drops per half liter of water, the ability to root increased by 75%. After 1.5 months of rooting and spraying with peptides, Petunia flowering become more active. Also, in early October, the Petunia mother plants were heavy pruned and transplanted from open grow into 1.5 liter flower pots. The plants were sprayed with Revilab or SL09 daily, and after that, the survival rate of Petunia after using of peptides complex was near 95%. Also, we can see the positive effect of Revilab SL09 on the growth of Alternatera and Verbena plants. We can do the scheme of the application of lingual peptides for improvement of growth, increase of stress resistance, and activation of flowering of indoor plants and seeds. At the first month, we can recommend use Revilab SL09 drops per half of liter water, and in the second month, we can recommend use Endoluten lingual, Vladonix lingual, and Taxaris linguals, all the 2 drops per half of liter of water. Prepare the peptide solution immediately before watering. Spray the plant leaves with a spray bottle with the peptide solution 2 times a day, morning and evening, plus water the plant with 50 milliliters of water with the peptides once every 3 days. After 1 or 2 months, the use of peptides can be repeated. Thank you for your attention.